transport chain. And we're looking specifically at the process of oxidative phosphorylation. Let's make a note here that this requires oxygen. We have our mitochondria. You know that it has this double wall. It has two membranes, an inner and an outer. And so across this area of the mitochondria is where we're kind of focusing. This is the outer membrane. This is the inner membrane. And this is the intermembrane space. Okay, the outer membrane, the inner membrane, and then the intermembrane space here. Now the first thing I'll illustrate is the relevant concentration of hydrogen ions on either side. So we've got a lot of hydrogen ions in the inner membrane space. So I'll just put a high concentration of hydrogen. Whereas we have relatively few hydrogen ions in the matrix down here. So this is the mitochondrial matrix. So we have a low concentration of hydrogen ions. We also want to point out the complexes. So the complexes are molecules that are sitting in the inner membrane. And the final most important complex here is ATP synthase. Okay. Now don't be too worried about the names of these complexes. Just importantly understand that the final complex is kind of where the most important step happens. So this is called ATP synthase. Alrighty. Okay. Now we know that we brought electrons in from the processes that we spoke about. So from the Krebs cycle, from glycolysis, we brought in, we had NADH with the electrons that it was carrying. We had FADH, also carrying some electrons, and that is going to be oxidized. So just to remind ourselves here that oxidation is loss of electrons, right? Remember that mnemonic, OIL, and RIG, reduction is gain of electrons. So we're going to oxidize these carriers. Oops. And we're going to strip off those electrons, and we're going to end up with the oxidized form of these carriers once again. And so now we've liberated the electrons, and those are going to go through the complexes. Okay. So we brought those carriers in their reduced form. We oxidized them by taking those electrons off, and those electrons enter the complexes, and they're basically going to be passed from complex to complex. So they're going to enter complex 1. And as they enter complex 1, we're going to pump some hydrogen ions through the first complex. So every time an electron is passed from one complex to the other, hydrogen ions get pumped back out into the intermembrane space. And that is why we have this high concentration of hydrogen out here and a relatively low concentration of hydrogen in the matrix. So once again, we're going to pass the electron, again, from this complex onto this complex. And every time we do that, hydrogen is pumped across the inner mitochondrial membrane and into the intermembrane space. And we continue to do that. Now, very, very, very important part of this process. In order for ATP synthase to pass that electron on somewhere else, right, there needs to be oxygen present. There needs to be oxygen present. So the whole purpose of oxygen is to be the final electron acceptor here. I'm going to draw one big oxygen molecule. Okay, go two in here. And oxygen is basically going to be the final acceptor of those electrons. And that will facilitate the flow of hydrogen ions from this high concentration up here into the matrix. And that is how we synthesize ATP. Okay. So we're basically going to phosphorylate, I'll draw it down here, plus a phosphate, use as ATP. Okay. So I'll go over that one more time. So we brought in these reduced form of our carriers. They were carrying those electrons from those other processes we mentioned. We oxidize them to free up or liberate those electrons. The electrons are then passed from one complex to the other. And every time we pass electrons from complex to complex, hydrogen ions are pumped through these complexes. Okay? Now, at the final complex, which is the most important, ATP synthase, we have oxygen taking the electrons from ATP synthase, and that is driving the hydrogen ions down their concentration gradient. So basically, these complexes were setting up a gradient. So by pumping all of these hydrogen ions out, we've set up a gradient so that when we, excuse me, so that when we pass that final electron down to oxygen, we can have hydrogen flowing down this gradient, past this ATP synthase passively because of that established hydrogen gradient. Okay, and as these ions flow down their gradients, we're going to talk about this type of movement later on, but when molecules move according to their concentration gradient, they basically liberate some energy to do work. So as the hydrogen ions flow down their gradient, they liberate the energy to add this phosphate onto ADP and to make ATP. Now this is a much more efficient process. This is where we yield most of the ATP in our bodies. I'm just going to make a note here. We make about 34 ATP per glucose. Whereas we know that the other 4 came from the Krebs cycle and came from glycolysis. So all of the 38 ATP that we yield per glucose, 34 of those ATP are coming just from the ETC alone. All right. Okay. Any questions here? What questions do we have at this point? Yep. Mm-hmm. Yeah, this is part of the NADH. It's carrying 2 electrons. Okay. Mm-hmm. So it goes from NAD plus to NADH2 plus, and then it's oxidized back to NAD plus. Okay, this is also carrying 2 electrons. So they're both carrying 2 electrons each. Any other questions? Any other questions? Okay, so really important to stress here that the whole reason that we breathe oxygen is to drive this process. And if oxygen is not here, then this entire sequence of events kind of halts right here. and we have the buildup of electrons, we have the buildup of hydrogen ions, we have lots of oxygen physically in the system, physically in the blood, but we basically have the signs of hypoxia because oxygen is not liberating the energy to make ATP. Okay. Really, really important. Okay, so this is basically just kind of reviewing those steps. Let's start by looking at the overall reaction down here at the bottom. So basically we got 10 NADH plus those 10 H's. So basically 2 electrons per NAD plus. And then we also have 2 FADH2 that is coming from the Krebs cycle. We have 28 ADP and 28 phosphates, and that yields us 28 ATP, 12 water, 2 FAD, and 10 NAD+. So back in the oxidized form. So the really important part here is that oxygen must be present to accept those final electrons. Okay, oxygen must be present to accept those final electrons. And I want to stress that we can make various amounts of ATP in the electron transport chain. Again, that's going to depend on how efficient we are, and that can change by physiology, that can change by our body state, and other things like that. Okay? But the big take-home message is that most of our ATP is made by this mechanism. Right, so let's just quickly run through the sequence of events here just one more time. We have those electrons moving through the complexes, and then when it gets to that final complex, we have oxygen standing in the way to grab that final electron, and then we have ATP synthase moving all of this hydrogen down its concentration gradient passively. So note this green arrow means that these are moving passively, whereas these red arrows mean that this is being pumped against its gradient. And then this movement of hydrogen passively down its gradient is where the magic happens of making that ATP. Okay. So to look at the big picture of putting all of this together, we had glucose yielding a little bit of ATP, that linking step yielding no ATP, but yielding some carriers, as well as some carbon dioxide as a byproduct. We had Krebs cycle yielding a significant chunk of those electrons, so FADH2 and NADH+. And then we also have some carbon dioxide and a little bit more ATP. And then down here at the electron transport chain, we take all of these carriers that are carrying electrons, we shuttle them off into this process, and we generate a significant chunk of our ATP.